We're going to power through, make it to the end. All right. For embryology. Okay, great, good. We're back on track. Okay, for embryology, I really want us to get to understand germ layers. I want us to understand a few components of organogenesis. And from there, that could be enough. If you are interested in this topic and want to go deeper, great. NPLEX seems to vary from having no questions to lots of questions in this section, but these typically count as embryology questions unless if they're more discussing fertilization, which is typically more physiologic. We're not going to talk about fertilization today. We are doing the talk about embryogenesis from implantation, gastrulation, and neuralation. So week 1, our blastocyst sticks on day 6. That's our implantation day. So from day 1, fertilization, moving through the uterine tube, becoming a two-cell stage, 4-cell stage, morula and blastocyst. When it started as a zygote, it then will implant by day 7, but it'll typically stick on day 6. Around this time, we'll see HCG secretion beginning. Then in week 2, that's when we have our separation into 2 layers, our epiblast and our hypoblast. We can see this 2-layer structure being formed as this is the blastocele. These are the embryoblast. Blastocele is a whole trophoblastic cells on the outside. So this 2-layer structure will eventually become our 3 germ layers. Our epiblast and then hypoblast, we can see this hypoblast down here as the separation of from the epiblast cells from the actual blastocele itself. We call these 2 layers the bilaminar embryonic disc, and we will look and see soon how this epiblast layer is going to, well, really the bilaminal layer is going to form our germ layer, our 3-layer formation, which we get 3 layers by week 3. Gastrulation is the process that forms our trilaminar embryonic disc. We get our ectoderm layer, which we'll see on top. The overlying ectoderm will eventually become our neural plate through neuralation. We get our mesoderm layer represented in these red cells. Here in the center, that's going to form our notochord, which will allow us to have a midline for the body for development of our mesoderm. And then we have this endoderm layer. This endoderm layer, will, as we'll see, will become a lot of our glandular internal organ structures, like our digestive tract. During embryologic development, again, what occurs on day 6, it sticks. We see implantation on the uterine wall. So our ectoderm is going to be our external and outer layer. We see our surface ectoderm becomes things ranging from our epidermis of our body to our anterior pituitary, the lens of our eye, the epithelial linings of the oral cavity and other things, the parotid, sweat, and mammary glands, and the anal canal below the pectinate line. So think all things, really kind of a lot of these external components, outer layer, surface-facing components. It also, though, forms our neural tube, and we call this neuroectoderm, and this is going to create our central nervous system structures like our brain, which includes our posterior pituitary, our central nervous system neurons, oligodendrocytes, our astrocytes, our ependymal cells, our pineal gland. So a lot of these supportive structures that support brain function, our retina, and our spinal cord. The neural crest as well is formed by the ectoderm. This is going to become our peripheral nervous system and other non-neural structures that surround. And so we'll look at what this neural crest means. So ectoderm is surface ectoderm, so kind of surface structures, the neural tube, which becomes our central nervous system, and our neural crest, which becomes our peripheral nervous system, as well as Elmo-Passes. That's the acronym I have, which is our enterochromaffin cells, our leptomeninges, so our pia and arachnoid mater, melanocytes, odontoblasts, which is the outer cell of pulp that secretes dentin, our peripheral nervous system ganglia, so those collections of nerve roots or nerve cell or actual neuron cell bodies, adrenal medulla, the inner portion of the adrenal gland that secretes your catecholamines, Schwann cells, which are a neuroglial cell in the peripheral nervous system, the spiral membrane, which eventually becomes fused with the interventricular septum of the heart, endocardial cushions, which are partially derived from mesoderm, which will create the walls to create the 4 chambers of the heart, and the skull bones. My goodness, lots of different structures. How to remember it? Sometimes it might be better to remember what it's not. So if you're able to understand kind of surface ectoderm, what these pieces are, the neural tube, central nervous system, and then know that the neural crest is the peripheral nervous system as well as some other stuff. Let's see what endo and meso are, and then we can maybe go back and place other things in this neural crest cell place. Mesoderm is like the middle or the meat layer of the body. So our muscle, bone, and connective tissue, the peritoneum, pericardium, pleura, our cardiovascular structures as a whole, our spleen, proximal vagina, wall of gut tube, blood, lymphatics, our kidneys, our adrenal cortex, dermis, testes, ovaries, and tracheal cartilage. So a lot of the middle stuff in the body. It also is going to form our dura mater. Remember, our other 2 layers are going to be from our neural cells and the notochord, which helps to induce the ectoderm to form the neural plate, which helps kind of things become aligned on a central axis. Eventually, the notochord becomes the nucleus pulposus, which is inside those intravertebral discs. And then our endoderm is our internal layer. This is where we have our gut tube epithelium, the lining of our gut. Which includes the anal canal above the pectinate line, our urethra, the majority, and the distal vagina. A lot of our epithelial derivatives, so a lot of these different organs here, a lot of them glandular in nature, like thymus, parathyroid, pancreas, but also other things like eustachian tube, gallbladder, liver, and lungs. And so when we look at these 3 kind of structural layers, this is actually pretty high yield, knowing who is derived from what 3 germ layers of tissue. So I'd say this is worth looking into and kind of putting in your brain a bit more. When we look at the actual process of gastrulation forming these layers, we see we have the bilaminar embryonic disc with our epiblast and our hypoblast, and then we have our trilaminar embryonic disc with ecto, meso, and endoderm. And to form kind of the beginning of organogenesis, we'll see that we start to create this tube with this notochord. But we get there. Epiblastic cells invaginate, kind of move inward through the primitive streak to form the 3 germ layers. And this is around day 15 where we see the primitive streak form. And day 16, we see the 3 kind of germ layers form. So we're still pretty close on in development. By day 19, that's we've gotten this notochord formed from our mesoderm. And that's really where we can start kind of orienting structures by day 19. So once we have this midline notochord formed, we can start the process of neuralation. And you can see this notochord is centrally located here. Neuralation is really the creation of the neural plate from overlying ectoderm, utilizing where the notochord is as the guide. So on day 19, our midline neural groove and bilateral neural crest, so groove, crest, regions form in our ectoderm. And it knows to form there because it follows along wherever the midline notochord is. From a genetics perspective, sonic hedgehog gene actually is the gene that induces this change. And so if you saw a mutation or an issue in neuralation, you could go back to maybe a mutation in this actual gene, this genetic component. This helps us create our anterior posterior axis patterning. It helps us do the process of central nervous system development. So if this gene has a genetic mutation, we're not going to be able to do the process of neuralation correctly. From here, the next day, day 20, we see the neural groove is really developing, it's deepening, and we actually create a full fold, and the neural crest cells will start to break off from the rest of the ectoderm and move deeper into the ectoderm layer bilaterally. And so we can see this not so great here. I'm seeing if we can see it in this page. No, I don't have your neural crest cells on here, at least breaking. Oh, yes, I do, kind of. Neural crest cells kind of breaking off here. You'll see them breaking off shortly once they form the tube. Once we have the tube fully fused together, day 22, that's when we say that the neural tube has been formed from the neural plate. And you can see now that the neural tube is completely separate from the other external ectoderm layer that will become like our epidermis and other external structures. But they both derived from the same embryonic germ layer tissue. And then the neural crest cells will lie kind of superficial to the neural tube bilaterally. And even though this is spinal ganglion, I would put these as neural crest cells right now. During week 4, we kind of are leaving neuralation for a second, because during week 4, we have the week of 4 as the heart starts to beat during week 4, because we get 4 chambers. And you can kind of visualize this cardiac activity then with an ultrasound. So that's why we can actually do an ultrasound by week 4 and see cardiac activity and hear a heartbeat. We also will see on our developing embryo upper and lower limb buds to actually form our 4 limbs. And this is just showing kind of our baseline development to get to these 4 chambers of the heart, showing this is the head, this is the tail at day 18, the cardiogenic area and primitive blood vessels. And we can see kind of blood flow through these different tubes, fusion, and then these kind of 4 different areas, truncus arteriosus, bulbus cordis, this primitive heart. Then by day 35, really a month in, we can see actually a real structure that starts to look like a heart. But by even sooner, by 4 weeks in, by 28 days, you can see we have these 4 chambers here, this partitioning of 4 chambers within this kind of truncus structure. So between here and here, we get this 4-chamber piece. Then by 8 weeks, we really actually have like the beginnings of what are going to be our valves. By week 7, we have the obliteration of the vitelline duct. The vitelline duct connects the yolk sac to the mid-gut lumen. We see this here. It's going to go away during week 7 of development because we've developed enough of these kind of primitive structures. And this image is kind of showing the difference between hindgut, mid-gut, and our foregut kind of up here. So again, the gene, Sonic the Hedgehog gene. Week 8, we have our genitalia formation. So the genitalia by week 8 have male and female characteristics. And that is our basic, like weeks 1 through 8, what's happening. I'm skipping through kind of the fetal development, like where we're actually like getting the extra structures. If you're interested in learning about the clefts, the pouch, these pieces, the pharyngeal apparatus is, you'll see cap, you'll have, which is outside to inside, you have clefts, which are the most outside piece, then arches, and then pouches. And so we can see in our clefts where they come from: ectoderm, arches, mesoderm and neural crest, and pouches, endoderm, and what they'll develop into. So we can see our clefts, and you can see these specific structures. I would say this is lower yield than your basic germ layers, but they have definitely been tested on before. So if you have the mental capacity to look through them, great. If not, that's completely fine. But they exist, and they're referring to those kind of components there. All right. We're going to go into organogenesis now. So looking back at the heart, I find that most of the embryology questions, if they aren't about germ layers, are often about cardiovascular structures. So heart morphogenesis, it's the first functional organ in vertebrate embryos. By week 4, it's beating spontaneously, and we'll see that there's left to right polarity that starts to establish itself during this time as well. So when we're separating into our chambers, we'll see that this septum primum, this green little tiny line, will grow toward the endocardial cushions. And the endocardial cushions, you can kind of see these structures here. And remember, those endocardial cushions developed from those neurocrest cells, as well as from mesoderm. Then we see the ostium secundum, which I believe we can maybe see in this picture, maybe not. But ostium secundum will form in the septum primum due to cell death. So you'll start to see this portion kind of regress. The septum secundum is going to develop on the right side of the septum primum. It'll maintain a right to left shunt, because remember, we aren't doing that full oxygenated pathway from deoxygenated to oxygenated, because our lungs aren't taking in oxygen when we're in a fluid sustained state. So we are still having a shunt from right to left side to allow blood flow to occur. The septum secundum will expand and cover most of the ostium secundum. The residual foramen is the foramen ovale. So we can kind of see these two formations forming together. So coming down and meeting upward, and then this space between, which will be our foramen ovale that's opened. The remaining portion of the septum primum here forms a one-way valve of the foramen ovale, and this will close, sealing the foramen ovale soon after birth. And this is partially because we have increased left arterial pressure and decreased right arterial pressure. So essentially we'll push back again this portion and it will close and fuse. Now it can remain open sometimes, and that's a birth defect that can occur. Oftentimes though, over time it will close just naturally. Sometimes surgical intervention needs to happen. Okay. Looking at the chambers, the separation of the ventricles, that was the atria. The muscular interventricular septum forms. The opening is called the interventricular foramen. The aortico-pulmonary septum will kind of rotate and fuse with our muscular ventricular septum to form this membranous interventricular septum, and it'll close off that foramen. And then growth of the endocardial cushions will separate the atria above from the ventricles below and contributes to that separation between the two. I don't think I have a great picture of that here. I might have one in the previous. Yeah, I don't have a beautiful picture. You can kind of see it in this image here. That endocardial cushion is expanding downward, and same as this one here, and they're eventually going to fuse, but that's that septum here between the two ventricles. Or that foramen between the two ventricles that will eventually become a closed septum. Our outflow tract. Okay, so I do have some of those pieces here. So from our ascending aorta and pulmonary trunk, we'll have a couple different, both the truncal and bulbar ridges will spiral and fuse to form, and they'll arise from neural crest cell migrations. The aortic and pulmonic valves come from endocardial cushions of the outflow tract, and the mitral and tricuspid valves are fused endocardial cushions of the atrioventricular canal. If we look at this picture here, we can kind of see how these structures are forming together, and see these special names: truncus arteriosus, bulbus cordis, ventricle, and then our sinus venosus or arterios or venosus, essentially the venous blood, but it's all intermingling here, right? We're having intermingling of arterial and venous blood until after birth. Our aortic arch derivatives, this is kind of like your pharyngeal pouch component or conversation. If you want to get into this and see like who is communicating with who here, who becomes what, go for it. I have it all here for you. So you can see our first arch is going to form part of the maxillary artery, which is a branch of the external carotid. So first arch is maximal. I tried to kind of make it a little bit more easier to understand, but I'd say this is about the same level of yield as the pharyngeal pouches. So if you don't have the capacity to put it in your brain, I would not put a lot of time into this. Okay. This portion is a little bit more important, unfortunately, mainly because I see these words, I see these structures mentioned more on NPLEX. So our truncus arteriosus, we can see this structure here. It's going to become the ascending aorta and the pulmonary trunk. So think truncus arteriosus as really the valves that are going to be leaving the heart, ascending aorta and pulmonary trunk. Our bulbus cordis, which we can see kind of right off that truncus arteriosus, is going to become the smooth portions of left and right ventricles. So bulbus cordis will eventually flip and become the smooth portions of left and right ventricles. Our primitive ventricle and primitive atrium are the trabeculated portions, the portions that have that trabeculae muscle on them that are lumpy bumpy. The left horn of the sinus venosus, so we have our sinus venosus here, the left horn of it is going to become the coronary sinus, which will drain back into the right atrium. The right horn will become the smooth portion of the right atrium that doesn't have pectinate muscle. Endocardial cushions, we can't see them here because it's inside the heart. Those are becoming our septum, so atrial and our membranous interventricular septum, as well as all of our valves as our endocardial cushions. The right common and anterior cardinal veins become the superior vena cava, The posterior, subcardinal, and supracardinal veins become the inferior. And then the primitive pulmonary vein becomes the smooth portion of the left atrium. You can kind of see like how these structures could give rise. I would say the most important structures in this slide set, my thing is getting stuck, would be truncus arteriosus, bulbus cordis, and endocardial cushions. I would know these three and what they give rise to, and then I wouldn't probably worry about the others. In fetal circulation, there are three important shunts that we need to kind of care about. Blood enters the fetus through the umbilical vein, and it's conducted via the ductus venosus into the inferior vena cava, allowing it to bypass hepatic circulation. So we can see this ductus venosus here, umbilical vein here, and then IVC here. It doesn't have to go through the liver. So this would be one of our shunts. The majority of highly oxygenated blood reaching the heart via the IVC is directed through our foramen ovale directly into the left atrium, therefore having to skip kind of this pulmonary trunk portion. And then deoxygenated blood through the SVC passed through the right atrium, right ventricle, main pulmonary artery, ductus arteriosus, descending aorta, goes this direction because there's pretty high fetal pulmonary resistance because we are in a liquid environment. We're not breathing into air. So we can kind of see this passage here of blood that's deoxygenated, that's moving down this pathway from right atrium, right ventricle, pulmonary artery, ductus arteriosus, and then descending aorta, because it's not going to come down here. There's a lot of resistance in this pathway here. So it doesn't want to go down this shunt. It skips kind of this pathway for the most part. And this makes sense, right? Ductus venosus, foramen ovale, and ductus arteriosus are structures that we don't see in adults. So it would make sense that they're going to be involved in shunts for fetal circulation. So that's how I remember the three. Ductus venosus is associated with our oxygenated blood from our umbilical vein. Our foramen ovale is the shunt communicating between the sides of the heart. And our ductus arteriosus is taking oftentimes deoxygenated blood out of the pulmonary trunk back out through to the placenta. If you're interested in lung development, I have it here. I don't think it's super duper high yield at this time, but if you are interested in looking at it, remember that piece of it is endodermal tissue. That's its germ layer. And this is the 5 stages of lung development. You could remember it as every pulmonologist can see alveoli for the 5 stages in order. GI development would be more important for organogenesis, so heart and gut. Our foregut is the structures of the esophagus to the duodenum at the level of the pancreatic duct and the common bile duct insertion, so the ampulla of Vater. That would be the foregut region. Midgut is post that, or post inferior to that, the lower duodenum to about the proximal 2/3 of the transverse colon. And some important things that happen in this section is week 6, we'll see herniation. It's physiologic, not pathologic, meaning it happens to every developing fetus of the midgut through the umbilical ring. And week 10, we'll see that it returns back to the abdominal cavity, rotates almost all the way around 270 degrees counterclockwise around the superior mesenteric artery to try to it's actually starting to form this like structure that's interrelated with this artery. And if we think about the fact that this is our small to large intestine, all the interweaving that that intestine does, it makes sense that we start to form these big flexures that eventually we'll see as our flexure, like our hepatic flexure over time. And then the hindgut is the distal 1/3 of the transverse colon to the anal canal above the pectinate line. Remember, that's the section that counts for this type of region. And all of these components, the lumen of our digestive tract and the epithelium is going to come from endoderms. And we can see up top here above this gut is where we have those pharyngeal pouches 1 through 4. I think that's probably your most high yield piece. You can kind of see some arterial intervention already starting to happen from our celiac, superior mesenteric, and inferior mesenteric. Neurologically, we've looked at this already, but to look at it again, our notochord is going to induce the ectoderm overlying to differentiate into neuroectoderm and form our neural plate. From there, we'll see the neural plate will create our neural tube, which is now fully here, and then our neural crest cells, these little green cells here, and then this will eventually become our epidermis. All of this, though, is ectodermal germ tissue. The lateral walls of the neural tube are divided into plates. They have similar kind of orientation of the spinal cord. We have the alar plate, which is dorsal and sensory, and basal plate, which is ventral and motor. The alar plate is induced by BMP, bone morphogenic protein. The basal plate is influenced by SHH, our sonic hedgehog gene. So if we look at these different components of the neural tube, we can see that in 4 weeks, 5 weeks, and then the adult wall derivatives and cavity derivatives. So 4 weeks, we have our primary brain vessels. We have our proencephalon, our mesencephalon, our rhombencephalon, and our neural tube. You could also break this down into forebrain, which is proencephalon, midbrain, which is mesencephalon, and hindbrain, which is the rhombencephalon. So F, M, and H here. We can see that the mesencephalon is going to continue to be the mesencephalon, and this will eventually become our midbrain portion of our actual brain stem. We will have the cavity associated is our cerebral aqueduct. Our forebrain becomes the telencephalon and the diencephalon. So these two structures here at 5 weeks. The telencephalon becomes our basal ganglia and our cerebral hemispheres, as well as our lateral ventricles. Our diencephalon becomes the thalamus, hypothalamus, and retina. So really our integration centers for a lot of that autonomic nervous system components and the third ventricle. The rhombencephalon becomes the meta and the myelencephalon. The meta becomes the pons and cerebellum, so the middle brainstem and then the cerebellum portion, as well as the upper portion of the fourth ventricle. The myelencephalon becomes the medulla, the major autonomic nervous system integrator, and has all of those centers, and that's the lower portion of the fourth ventricle. So this is kind of our personalization of the neural tube. We can look also at the kidney. Again, I would say kidneys lower yield, kind of closer to the lungs, but you could be asked about these three pieces: the pronephros, the mesonephros, the metanephros. The pro week 4 and meso week 4. The pronephros degenerate. The mesonephros is kind of the interim kidney at the beginning, and then the metanephros is going to be your permanent kidney, and your kidney development is complete by week 36, so 4 weeks before birth. Some other components of nephrogenesis. And then a big piece, this is a little physiology, but I'm just going to touch on it, is the young liver is who's synthesizing your blood. Weeks 3 to 8, your yolk sac's doing it. 6 weeks to birth, your liver takes over. And then 10 to 28 weeks, your spleen. 18 weeks to adult is your bone marrow. If we look here, we can kind of see there is crossover, right? So from fetal hemoglobin to adult hemoglobin, alpha is always there, gamma goes and becomes beta. What does that mean? It means that adult hemoglobin is going to have 2 alpha chains, so alpha always, and 2 beta chains. So that's your adult hemoglobin. Remember, it's a 4-protein quaternary structure, 4 polypeptide quaternary structure. Fetal hemoglobin, on the other hand, is going to have 2 alpha chains, alpha always, and 2 gamma chains, and gamma is going to go away, right? So this is only fetal hemoglobin. When we look here, we can see kind of a couple different pieces, like who's doing what. Our yolk sac is really active here, then our liver starts to take over. The liver will last pretty much all the way until, almost like months of just a past gestational stage. The spleen helps out in the middle, and then bone marrow ramps up. So by the time birth happens, right? By the time birth happens, we see a little liver, a little spleen, but our bone marrow is almost taken over control of creating our blood erythropoiesis. And then as into adulthood, obviously bone marrow is the strong one. And that's why at birth we see this beta chain really increasing, and our gamma chain is just going away. Alpha is always, so alpha is constant. All right. So with that, I'm going to close down my screen and look at your faces, check in, and talk about what would be good next steps with all of this anatomy thrown at you today on a Saturday snow day. My suggestion, so we've done biochem. Hopefully you're still kind of going through biochem practice problems, reviewing those pieces. Layering in anatomy is a good break for your biochem brain. I would do coloring books if you're needing more moderate yield, if you need more of a broad review, coloring books and practice questions. I would do fill in the blank images if you're also kind of in that moderate yield category where you're not able to visualize the structure in your brain yet. Use a fill in the blank picture where you can have the picture there unlabeled and you label it yourself. From there, after this next week, my goal for you with about 2 to 3 weeks left is to transition to being able to either draw out the structures yourself in kind of a schematic or flashcards where at least the initial flashcard or practice question doesn't give you an image. And then when you answer it, you can see an image after for confirmation. But that's really should be moving into over the next 2 to 3 weeks, maybe after this next week of self-study. Okay. That's what I would focus on. We're going to go into micro and immuno next. I don't need you to do any prep work for that. We will do a practice exam next Friday, hopefully in person. If not, I'll have it available for you electronically, but I feel like we'll be back in person next weekend. And then next weekend, we'll spend about 2/3 of our time on micro and about 1/3 of our time on immunology. I'm going through some high yield pieces. And then the last week will be your physio path. And then you'll be free to take the exam and hopefully put this behind you forever. Moving forward. Yeah. Any supports that you already haven't requested that I can do? I've already heard supports for additional practice questions and additional practice tests for anatomy, which I can provide, so I can work on those and to make sure the videos get uploaded. So those 3 pieces I can do. What else do you need from me?
I had a question about erythropoiesis. Does that take place also in the kidneys, and what would be the primary organ, I guess, that makes erythropoiesis in adulthood? Would that be bone marrow only?
So are you talking about not hematopoiesis, not the creation of our red blood cells or erythropoiesis, but you're talking about erythropoietin, the hormone, or are you talking about the process?
I guess all. I'm
All
just
of the above. Yeah, totally. That's fair.
Okay.
So the process of erythropoiesis is just the process to make red blood cells, erythrocytes. And that process is typically governed by bone marrow, right, in adulthood. And then as we are growing through the fetal stage, you'll see yolk sac, liver, spleen, and then bone marrow taking over. Erythropoietin, I think where you're coming in is erythropoietin is produced by the kidney. It's secreted by your kidney. And so that's the hormonal influence of do we need more red blood cell creation or not? We don't need more red blood cell creation. And the kidney is who is actually signaling that we need more or less. And that's secreted from the peritubular cells of the kidney that stimulate red blood cell production. But they're not actually producing the red blood cells in the kidney. They're just stimulating the production from the bone marrow. So think of erythropoietin as the hormone secreted by the peritubular cells of the kidney when we have the sensation that our blood counts are too low. EPO then will go act on bone marrow to increase erythropoiesis.
Awesome. Thank you.
Yeah, super good clarification point. I love that. I think this is a super, here, let me just put this picture